Hello students, this is Faizan Mirza. We are discussing monoclonal antibodies. Monoclonal antibodies are one type of antibody which are produced on commercial scale, on a large scale, in a lab or in an industry. Plasma cells from mice are used, and these plasma cells are those which can secrete the antibody against the antigen which we are interested in. And these cells are collected from the spleen of the mice. Plasma cells do not divide. Although they are producing antibodies, they do not divide. So we make hybridoma cells, which are produced by fusing the plasma cells with myeloma cells. What are myeloma cells? Myeloma cells are cancer cells which have infinite ability to divide. So when we have hybridoma cells, we have these cells, we produce clones of these hybridoma cells. These hybridoma cells will keep on dividing by mitosis again and again and again. They will divide continuously, and since they are made by fusion with a plasma cell, so they will have not only the ability of dividing continuously, they will have the ability of producing one type of antibody in large scale. These cells are grown in the tissue cultures in the lab, and the same monoclonal antibody can be produced on large scale, which we actually wanted. This is the mode of, you can say, the protocol which is followed. We take a mice, we inject the antigen in the mice. So when the antigen is injected in the mice, this is the antigen against which we want the mice to make the antibodies. So we inject the antigen in the mice. The B cells in the mice will recognize the antigen, they will proliferate, they will differentiate, and they will accumulate in the spleen, and plasma cells will form. We take out the spleen, and the plasma cells are isolated from the mice. They are screened for the plasma cells that we want. These are taken out so that we know that these are the ones which are producing the antibody against this antigen. These are the cells which we are interested in. Now we take myeloma cells or cancer cells, we fuse both these cells together to form the hybridoma cells. The hybridoma cells will have the ability of producing the antibody in large scale, as well as they keep on dividing. So these cells are cloned and they keep on growing. This is how the monoclonal antibodies are made. Let's discuss the application of these monoclonal antibodies. For example, trastuzumab. Trastuzumab is a medication which is used for the treatment of breast cancer. The name ends at mab. Mab means monoclonal antibody. It is used for treating breast cancer. So you know that you can recall the structure of an antibody. There are two antigen binding sites, and then there is a constant region which can attach to the neutrophil. So the neutrophil will have the receptor for this constant region, and the antigen binding site will be complementary to a specific antigen. So trastuzumab is used in the treatment of breast cancer. So what's the application of monoclonal antibodies? MAB, MABs, they are used in treatment and they are used in diagnosis. How is it used in the treatment? So for example, this is a breast cancer cell, and this is a normal cell of the same breast tissue. And this is the neutrophil. You can see the lobe nucleus, and this neutrophil will have the receptor for binding to the constant region of the antibody. Now, we have made an antibody which is complementary to this antigen which the breast cancer cell is displaying, this orange dots. These are the antigens, and these antigens are present on the breast cancer cell in a very large quantity. So the antibodies will go and attach there, and when they go and attach there, these antibodies, the constant region will be the one which will be complementary to the receptor present on the neutrophil cell surface membrane. So the neutrophil will recognize these, and they can go and they can just evoke an immune response against this particular breast cancer cell. The normal cell will also have the same antigens, but in very less quantity. So the same antigen is overexpressed in a breast cancer cell. So the breast cancer cell is highlighted more as compared to a normal cell. This is why the neutrophil will go and it will target it. It will just target the cancerous cell, and it will just make sure that the cell gets wiped out rather than just damaging all other cells of the body. So the targeted attempt can be made at killing the cancerous cells and not the normal cells of the body. Another process that we can do is in the diagnosis of deep clots in the body. An antibody is made with a radioisotope attached which can emit gamma radiations. So this antibody is made, it is raised against fibrin antigen, and this monoclonal antibody is an anti-fibrin antibody. What you can do, you can just inject this antibody in a patient who you suspect is having a deep clot in some tissue. So this antibody, this antigen binding sites here, they will go and they will attach to the fibrin present in the clot of that patient. no matter where the clot is present, no matter wherever how deep the clot is, these antibodies will have the antigen binding sites complementary to the fibrin. They will go and attach to the fibrin. Once that they attach to the fibrin, you can just place you can just note down the gamma radiation being emitted from the particular body part of the patient, and this radioisotope will keep on emitting gamma radiation. So you can just see the localized emission of gamma radiation from that patient, so the location of that deep clot can be actually seen. So these are the applications of monoclonal antibodies in the treatment and the diagnosis of various disorders. Thank you so much.